humans, which work as neural integrators. So neural integrators are like literally like mathematical integrators. So this is one illustration of neural integrator. What it does is it takes the pulse of a velocity signal, so any velocity command that goes into the integrator, it takes that command and then it converts it into the step. So literally it converts the velocity into the position, right? And he described that for the eye movement. So he described that neural integrator for eye movement take the eye velocity signal, which is saccade, for example, into an eye position signal. And the way he demonstrated that, he demonstrated it in the monkeys and cat brain. So this is a very classic old video that we got from David Robinson. What you see here is this is an oscilloscope, the classic old style oscilloscope. And what you're seeing, these grids are basically showing the position of the eye. So the grids on the vertical is vertical eye position, horizontal is horizontal eye position. I'm going to play this. As I play this, what you will see is this little bright dot, which is the position of eye. It will move in different directions. And as it will move in different directions, you will hear the cells that he has recorded from the midbrain vertical neural integrator, and those firing changes. So you see here. So what you see is it's like a humming bee, right? So it kind of like is much louder and much frequent when the eye is towards the bottom, and it kind of silences down when it goes on the top. What that means is that neural integrator here is responsible for downward eye movement neural integration. And that's where he recorded from. Yeah, and he also demonstrated that in monkeys, when you remove cerebellum or you disrupt their vision or orbital proprioception, then this neural integrator gets dysfunctional. So as engineers, I don't need to tell you that any integrator is not always perfect. There is always some leakiness to the integrator, right? So it does not stay steady like that, but the output of the neural integrator leaks over a period of time. And that leak in the neural integrator, which is absolutely not acceptable for the brain. So what brain does is it gives the feedback in form of different sources, which corrects the leak, and that kind of improves the fidelity of the neural integrator. And it turns out that it's cerebellum, vision, and orbital proprioception. And if you remove these things, any of these things, then you will see the leak. So few years later, after Dave Robinson described neural integrator, Dave Z started working with him, who was a neurologist. And he published this paper in Brain in 1976, much older than many of us are, where he showed that if you take patients with cervical cerebellar ataxias who have damage to their cerebellum, and if you look at their eye movements, the eye movements are suggestive of signs of leaky neural integrator. So, for example, in this patient, when she looks to the right, her eyes are drifting back to the center, and she makes a little correction. So these are kind of like a jerky movements like this, and which is a sign of neural integrator leak. In neurology, we call it gaze-evoked nystagmus. So if you record their eye movements with oculography, if you plot the horizontal eye position on y-axis and time on the x-axis, so what you see is when patient is looking straight ahead, there is no drift. It's relatively steady eye gaze holding, but when patients look to the right, it drifts to the left, corrects, drifts, corrects, drifts, corrects. And when patient goes on the left side, the drift direction reverses. So based on that, Z described the signs of the impaired neural integration in human disease or in non-human primate models is slow drifts of the eye, which are towards the center. These drifts are followed by fast corrective movements, which brings the eyes back to the target. There is a null position, for example, here, which is straight ahead, and there is no drift. And when eyes go opposite from the null, it drift direction changes. And not only that, the drift velocity increases gradually as eyes go farther away from the null. So these are like some rules that brain follows, which are sign of neural integrator failure. And these are kind of quite well embedded into ocular motor and neuro-ophthalmology field. And it's very well-known fact for many decades. So what happened is few decades later, some of our colleagues had out of orbit idea. So what they thought is we always talk about neural integrator with eye movements, but Ellie Claire, who's on the right side, she was doing her PhD with Doug Crawford in Yorks. And they asked, can we really apply this concept of neural integrator outside of the eye movement, let's say in the head motor control? So Doug, in his PhD with Tudis Willis, who was in London, Ontario, many years ago, discovered that for vertical eye positions, the neural integrator is in nucleus of Cajal. So with his student, then Doug asked, is that nucleus of Cajal also the integrator for the head movements? Because, you know, when you move your eyes, you move your head together, right? I mean, the eyes and head movements are very coupled movements. So intuitively, the integrator for the head should be somewhere near the integrator for the eyes, right? So what Ellie did is Ellie injected mucimol in nucleus of Cajal in monkeys. So what she saw drifts in the head movements of the monkeys after injecting mucimol in the Cajal. And then she published a paper in Science in 2002 describing the neural integrator for the head movement. Now, the same Ellie, after she finished her PhD, she moved to WashU, and she was my co-postdoc. So, of course, like Ellie and myself and my wife and many of our other colleagues, we all go for dinner together on Fridays. That was our tradition. And on our dinner, we don't discuss anything but about our monkeys. That's our discussion. Ellie was describing her monkeys and how their heads were moving around. I graduated from WashU. After that, I went to Baltimore. I was working with two people, David Z, who is a neuro-otologist, eye movement guy, and Buzz Jinnah, who is a movement disorder. I went to Buzz's clinic one day, and I saw this man who had some interesting head movements. So when he was moving his head, and I'm looking at this person's head, I was not a neurologist back then. I was a neuroscientist. And I thought like, this is like Allie's monkeys. I mean, these movements are like Allie's monkeys. So I called it head nystagmus. And of course, it brought humor out of my movement disorder neurologists because they never used the term head nystagmus. What they call this, they call it cervical dystonia. It is the third most common movement disorder affecting 3 million people worldwide. But based on this, although it's so common, they don't know why cervical dystonia happens and why these patients move their heads the way they do. So what we asked is cervical dystonia a disorder of head neural integrator? So what we did is we took patients with cervical dystonia and we measured their three-dimensional head movements. We did the exact same experiment that Dave Robinson and Dave Z did in 1970s, but instead of measuring their eyes, we measured their head. What we found, so when patient was looking to the right side, here, as you see, we are measuring his head movement. So there is a drift to the left, there is a correction, there is a drift, correction, drift, and on. These drifts are then minimal in one orientation. So in this particular case, it is minimum at 20 degrees to the left side, but the velocity of drift, as you see, is reducing as it goes to the left side. And when you turn his head farther away, when to the left, the drift direction changes. I'm not showing it here, but it does happen. So what we concluded is that cervical dystonia, the head movement in cervical dystonia are consistent with what we see as a result of dysfunction of the neural integrator. Therefore, cervical dystonia is a disorder affecting neural integrator. So this idea came in 2006. Of course, first time when I presented it, everybody laughed at me. I tried to publish it in Brain 3 times. It got rejected 3 times. 2 reviewers loved it, 1 hated it. We tried it 3 times. It got rejected. Mark Harrett was the chief editor. He told me, sorry, I can't do anything more. So then, like a few years later, we did some extra experiments, and we eventually published it in Journal of Neuroscience. So now it was getting more popularity over 5 years. But at the same time, I wanted to kind of get into more details of it. So what was the natural next step? It was to characterize the activity of the midbrain neurons in patients with cervical dystonia. Of course, we don't go with an electrode into patients' midbrain with cervical dystonia in today's age. So it was, of course, not the case. But then I came across this paper, which was published in 1972 from Japan. What they did back in those days, before botulinum toxin was invented, before DBS was invented, what people used to do is they would go in nucleus of Cajal in humans, and they would lesion the nucleus of Cajal to treat what they call torticollis, which is a name for cervical dystonia. I thought, like, gosh, I should be like 30 years older than what I am to do this thing. But of course, that was not a possibility, right? So I was lucky. I was lucky to find Alexei Sedov. So I met Alexei in 2015. He was presenting a poster on historic database from nucleus of Cajal ablation in 70 patients with cervical dystonia. So historically, before DBS, the ablation surgeries were relatively popular in Japan, in Germany, in Europe, and also in Russia. And Alexei was in the institute where they used to implant, or they still do a lot of implants, but they used to do a lot of surgical treatments for cervical dystonia. His PhD mentor was a part of these surgeries, and in his lab, they had access to historic database of a single units recorded from the nucleus of Cajal before they went in an ablation. So in any ablation surgery, with the way we normally do is we go in with a single unit electrode in patient's brain, we map out the activity of the area to be ablated, and once we know the coordinates of those areas, then we do the ablation, right? So in order to map out, we have to collect single units from those areas. We have to characterize their firing properties, their physiological properties, and whatnot. So what Alexei had, he had 52 neurons, which were recorded from human nucleus of Cajal, and they were all saved on magnetic tape. Not only the neurons, but it also had classified into eye-only nucleus of Cajal neurons, head-only nucleus of Cajal neurons, and eye-head. So remember, I was telling you that any integrator, so Cajal, Doug Crawford showed that it is the integrator for eye movement, and Ali, his student, showed its nuclear integrator for head movement. So of course, you expect the eye-only cells and head-only cells, right? But eyes and heads are coupled, so there are also cells in Cajal which are eye-head combined neurons. And we know these from monkey nucleus of Cajal, that these 3 neurons exist in monkeys. But what Alexei found, or his mentor found, and we published further, is that human nucleus of Cajal also had eyes, head, and eye-head combined neurons. So we had these cells. Not only that, we also had neck muscle EMG. So as we were recording the single unit activity, he also had the recording from the neck muscles in form of EMG. And of course, dream of an oculomotorist, he also had the oculography, which was recorded from patient's eye movement. So we had objective measure of eye movement, objective measure of neck movement, and corresponding activity patterns of single units, which was all saved on the tape. So what we did, we went back and we reanalyzed all the cells which are recorded on the tape to ask two specific questions, answering a very specific hypothesis. So the hypothesis is if a nucleus of Cajal neuron have signal for impaired neural integration of head movement, then the firing rate would decay with a specific time constant, right? And in contrast, we know that eye movements are normal in these patients. So the eye movement sensitive Cajal neurons will have a normal decay time constant. So in other words, the head time constant will be less than the eye time constant. This is an example of eye-only neuron, and what you are seeing here is, of course, this is cut from the tape, so it's not the best sound quality that you will see, but what you see here is its firing rate is much less. It is increasing as patient is asked to look up, and it is decreasing as patient is asked to look down, right? So but most importantly, what you are seeing, that eye-only neuron firing rate is persisting, right? So there is a persistent firing rate. In contrast, for the head-only neuron, there is no persistent firing rate. So what you can see here, there is a baseline firing rate, and when patient is asked to look up, it goes up and then it decays down. For some reason, I'm not able, but you got a point, right? So some reason I could not pause the tape. So there were two key predictions, right? So impairment, if impairment is intrinsic, so of course, so now we know that the neurons have signs of leaky neural integration, right? So then the question is that really intrinsic to head movement sensitive neurons and nothing to do with the eye movement sensitive neuron? Or in other words, there is a lesion which is a cell specific. It only affects the cells which are responsive to the head, and it spares the eye sensitive neuron. Or there is something wrong with the feedback, because feedback sources for the eyes and head are completely independent, right? So is that something to do with the feedback? So that's a second question that we ask. So we leverage the ability to have eye head sensitive neuron, right? So what question is that if the problem is specific to the eye or the head neuron, then what you will see, so if problem is with the integrator, then what you will see is if neuron is activated with the head or with the eye, it would decay, right? The eye head neuron, right? If the problem is with the eye head neuron itself. But if the problem is with the feedback, then the same cell will have persistent firing rate if the patient is moving the eye, but if patient moves the head, then there won't be persistence in the firing rate, right? So these were two specific questions that we asked. And what we found, the eye head sensitive neuron, so this is the neuron which is firing rate, time, it goes up with the head movement, and then it decays gradually in an exponential way with the head movement, right? But the same neuron, when you activate the cell with the eye movements, its firing rate persists. So what that means is the cell actually behaves differently depending on which feedback that is utilizing. What that means is that leakiness of the head movement neuron that we saw in dystonia patients is not really the problem with the brain, but the problem with the feedback that goes into the brain. So that's what we found. And we had several neurons like that, and we measured their time constant. And as you can see, that eye time constant was consistently higher than the head time constant, which suggests that for most neurons, the problem was the feedback, not the neuron itself. So with all this, what we could tell is that in cervical dystonia, the problem was with head neural integrator. The problem was not the integrator itself, but one of the sources of the feedback. Now, in dystonia, these sources of the feedback are the proprioception that comes from the neck muscles, the cerebellum, and the basal ganglia, right? So what I did is, and also these sources of feedbacks are also connected with each other, right? So the first thing that what we did is we vibrated normal people's neck. And then by vibrating normal people's neck, we asked whether we can create the dystonic eye movements in healthy subjects. Of course, the first normal person, hopefully normal, was me. I set into the search call system. I vibrated the back of my neck like this. So I vibrated the back of my neck. We mounted a little laser on my forehead, and I was supposed to turn my head and look around like that and keep my head in a dark to align the laser on the target, right? While vibrating my neck. So what we found is we found that my neck moves, but involuntarily drifts back and then it keeps correcting. So I had this dystonic type of movement of my own head that I appreciated when I was doing that. When I told that to Dave Z, he did not believe it. So I put him in the system, same thing happened. So, we did about 20 healthy subjects. Everybody had the same type of thing. And when we record their head movements with the search call system, this is what we found. So when they're looking to the right, they have the drift, which are to the left, and corrections, and opposite direction, the drift direction changes. And if you plot the drift velocity as opposed to the head position, drift velocity robustly modulated according to the head position. So this is classic. When we disrupted somehow the neck proprioception in normal people, we could create the dystonic phenotype in normal people. So then we ask, like, what happens if there is a decay which is coming out of the head neural integrator here? Then that decay not only goes here, but it also goes to the feedback. And if basal ganglia is part of that circuit, then we should see the decay in the basal ganglia neurons also. So now, 5 years ago, we started collecting this data, and I think we published that in 2017. And what we found, we recorded single unit from the cervical dystonia patients, their basal ganglia from their GPI, and we expected that these cells should also have the decay. So what we find, so this is a neuron, this is the firing rate, which is in gray. The EMG is in red. And this is the raster for GPI neuron, GPE neuron, and this is nucleus of Cajal from a long time ago. So what we are seeing is that GPI and GPE neuron, they both decay, and the decay is somehow parallel to the EMG, just like the nucleus of Cajal. I'm not going to go into the panel D figure, but if you focus on panel E, the time constant of the decay of GPI, GPE, or nucleus of Cajal is pretty comparable. So what that means is that whatever property we found in the leakiness of the head neural integrator, we also found that into the leakiness of the basal ganglia. So then what we ask, like, if I vibrate the neck of people with cervical dystonia, do I see any changes in the basal ganglia? Because this is connected, right? So if I change anything in the proprioception, can I capture that response from the basal ganglia if the feedback is connected with each other? So what we did, we recorded the cells from the basal ganglia. This paper, we are working on publishing this paper right now. So it will soon come out. But as we are recording, we vibrated their neck. So what you see here in this table, these are, I think, 152 or more neurons from GPI and GPE. And these are different firing parameters in response to neck vibration and without neck vibration. So what you see is that vibration does not really instantaneously change the firing rate, but it does robustly reduce the CV, coefficient of variance of firing, and also the frequency variance. In other words, the firing behavior, the regularity robustly changes as you vibrate, and that happens rather instantaneously. Remaining properties remain unchanged. And what we've also did in these 18 or 20 cervical dystonia patients is we measured the local field potentials from the recording electrodes, and we also saw the changes in the field activity. So this is the spectral power, and this is the frequency of the field potential. The light blue is without vibration, and red is with vibration. So, and this is like average of all patients. So the changes that I'm about to show you were much more robust in some and not so in the others, but there was obvious population change. So you see that little blip over here, which is not present in rest. So there was a significant blip into about 10 hertz or so field potential range, which was into the alpha band of local field potential. And when we measured the O scores of alpha band, the alpha band O score was significantly higher in those who had vibration as compared to the rest. So then we asked, like, how do we explain these effects of vibration in the basal ganglia property? Like, how does really vibration changes the basal ganglia activity? Is it a direct feedback? Does it come to the cortex, or it comes to the cerebellum? Is there a thalamic influence? We did not know. So the next question that we asked, we took help from this paper, which was published in 2015, where they characterized the functional connectivity with frequency-specific measure and coherence. So they measured basically magnetoencephalography and local field potentials in human pallidum. And what they found, they three frequency-specific networks. So one was a pallidotemporal network, which was a theta band, 4 to 8 hertz, which was connecting pallidum to the globus pallidum to the temporal cortex. Then second one was somatosensory, which was more into the beta or sensory motor areas of the cortex. And the third was alpha band, which is the change where we saw in our data with vibration. And interestingly, it was into coherence with the cerebellum. So alpha band, which is in coherence with cerebellum, did change with vibration, not the other bands. What that means is by vibrating the neck, we are actually modulating the circuit which goes through the cerebellum, and it changes the activity of the basal ganglia. Now, what all this means in a big scheme. So in big scheme, what we do right now for treatment of cervical dystonia is we basically either stimulate with DBS into the pallidum, standard of care surgery, or we inject botulinum toxin in patients' necks, right? So we change one of these two network and we change their calibration of the feedback, right? That's what we think is happening. But if they have stroke, for example, in basal ganglia, then what will you do? And if they have generalized dystonia, then you can't even do botulinum toxin. So what we could potentially do is we could do some kind of other measures, or you could find a different target, like cerebellar DBS to treat cervical dystonia, or modulation of the pathways where the cerebellum projects to. In fact, like I have about 3 patients who had dystonic neck tremor, and we implant, but tremor was so robust that it was almost like essential tremor. And in those patients, Jennifer Sweet and I implanted in the area, which was a little deeper from VIM in thalamus, where the cerebellar receiving fibers go in. And in fact, their neck tremor got better. So it fits with this theory. So we did not implant right in the cerebellum, but the outflow of the cerebellum, which goes into the thalamus. So it's basically the same conceptually. So this is all I wanted to tell you. I'm just going to project this circuit just for you guys to think. I'm not going to talk much about it, but this is the circuit that we had worked on, and this is a circuit that we used, like neural integrator connected with the cerebellum. Cerebellum gives estimate of the orientation and feeds back there, and the copy goes to the thalamus and then to the basal ganglia, back to the neural integrator, neck muscle connected to all this. So it's a network. So I think we can open for discussion because I'm done.
Thank you, Aasef. Very nice. Very nice. Nice story. I met David Robinson when I was interviewing for graduate schools. That was my only interaction. Did you know him?
I only got to meet him once a year in Crab Party, which I really want to start in Cleveland. So David Robinson was fan of crab and Maryland crab, right?
Yeah.
So Dave Z used to host this summer party where he invites Dave Robinson. All Dave Z's fellows stand in line, and we get 2 minutes to talk to David Robinson while he is not eating crabs. So, so 2 minutes every year. And last time I met him, it is an unfortunate story. One day before he passed away, he was admitted to Hopkins and the hospital, and I happened to be there, so I went to see him with Dave Z. But he was really sick at that time. But he had influenced everybody in our
He
field.
was very influential early biomedical engineer, no doubt. All right, Carl.
Hi, indeed, very
Hi.
exciting and interesting. And I'm sorry I joined late, but from what I saw is that obviously there's a challenge in collecting enough neurons. So the sample size of single neurons must be on the low side, but luckily you have the local field potential, and you can definitely bridge the gap here in potentially linking the spike to field and spike field coherence. And so the argument there is that if no matter how many samples you have, let's say 5 neurons or 6 neurons, if they all are coherent with the local field potential, you can make the claim that this is a representative sample of the total population because they're coherent with the LFP, therefore bridge the gap. But in general, the LFP data seemed interesting.
So Carl, we have a lot of LFP data we are working on right now, and we can collect even more now with Medtronic leads. I want to correct you, like, yes, we did have less neurons. We had only 52 neurons from nucleus of Cajal, but that was only because that data was collected in between 1975 and 1981. I mean, those surgeries stopped after 1981. So we did not have any more cells from that. But our last paper that we published, it has 750 cells from the pallidum. So we had like hundreds of cells. So, and we could do really good machine learning analyses and a lot of interesting things. I will send you the papers. Yes, that's correct. Like, it's hard to get cells during the surgery. But luckily we did not have that challenge because we had several neurons. We did do local field potential. Yeah, and of course, what you can do in the OR is very limited, right? So there are only certain things that we can do in OR when we can record cells. So it's always good to have a surrogate marker of the cellular activity, which is LFP, because we can collect LFPs outside of the OR. And that's what we are doing now, that we are doing coherence and using LFP as a surrogate marker in the same patient. And we are doing the experiment outside of the OR using local field potentials. That's altogether a separate talk, and I didn't really have a lot of time to put all data, but this was just part of it.
Okay. Well, that sounds even more interesting. And then the follow-up question is, rate coding obviously is one of the communication means between neurons. There's temporal coding, which gets a bit more complex. And you would wonder what the millisecond to millisecond behavior of these cells are, in addition to rate increase or decrease. Again, there are temporal pattern that could be changing there. But you can spend the next 5 years characterizing the populations within a nucleus, understanding the relationship to the LFP, and also understanding and maybe leveraging AI, as you said.
Yeah, so we have about 30 parameters that we use for the birth cell characterization. I didn't show all of them just for the interest of time. But there are, like you said, there are a lot, just not the firing rate, but the patterns. There are so many different patterns, and it's limitless. And there are so many different things we could do with local field potential. And in fact, we have been working already on 4 years on this. So 5 years is, I think, less. We need probably maybe 15 years to do all that. But yeah, we are looking into it, and we'll talk more with you about this. Sure.
Yes, I'll be very interested, and also see if we can offer anything on the preclinical mouse side with optogenetics. But irrespective, happy to chat.
Great.
Questions for Aasef? It was very interesting to see data taken off of magnetic tape. That's a really good use of existing data. It's really cool. Yeah, I always think people have a lot of data and it goes to waste. They finish the paper they're doing or whatever, and there's a lot of other potential uses for it. So fill out your data sharing plans and share your data.
Yeah.
Questions from the field? Any of our guests from Moscow? Okay.
Great.
Well, Aasef, thank you very much.
Yeah. Thank you.
Sorry. I guess that's it.
Great. Thank you.
Thanks
Yeah.
again. Everyone have a good evening.
You too. Bye-bye.